Hello friends, today we are talking about hemoglobin, which undergoes a structural change in binding to oxygen molecule. Okay, so after X-ray analysis, we have two major conformation in which hemoglobin exists, that is R state and T state. Okay, R simply stands for relaxed state and T is for tense state. And the point is, the oxygen can bind to both of them. But the thing is, the oxygen binding to R state, okay, it makes it more stable. If oxygen binds to R state, it makes it more stable. And if we talk about T state, then T state is more stable when the oxygen is absent. Oxygen is not there. So that is the deoxyhemoglobin. Okay, when the hemoglobin is present in T state and oxygen get bound to it, then it is converted to R state. Okay, but this conversion triggers a lot of changes, structural changes in hemoglobin. The binding of oxygen causes the heme to assume a more planar conformation. So moving next, we are talking about the cooperative binding of hemoglobin to oxygen. So we all know that hemoglobin transport oxygen from lungs to tissue. So here we will discuss why only hemoglobin is best suited for this function. Because we all know that there are number of proteins that have high affinity for oxygen. So like myoglobin, that have a really good affinity for oxygen molecule. So why don't we use myoglobin instead of hemoglobin? So the answer is, uh, here I have drawn a curve between theta and partial pressure of oxygen. Theta stands for the fraction of ligand binding sites that have been occupied. So the curve here I'm getting is the hyperbolic curve, that is for myoglobin protein. Why hyperbolic curve? Because myoglobin is a single subunit protein. Okay, and for hemoglobin it has four subunits, and it undergoes a transition from T state to R state that we have already discussed about. So this transition gives it a sigmoidal curve. This is a transition from low state to high state. So this type of sigmoidal curve is best suited for this function. And moreover, a protein that bound to oxygen with high affinity, we are not looking for that it will bind with high affinity. We're looking for a protein with a sufficiently low affinity to release the oxygen in the tissue, and it would not pick up much oxygen in the lungs. So that is hemoglobin only. So it explains why hemoglobin is best suited for this function. Moreover, hemoglobin is more sensitive to the small differences in oxygen concentration, because we can see that the partial pressure of oxygen is high in lungs and in tissue it is low. So it is hemoglobin is very much sensitive to the concentration of oxygen, so it picks up the oxygen from lungs and it releases into the tissue. Next we are talking about the allosteric protein. So firstly we will define what is an allosteric protein. Allosteric protein is in which the binding of a ligand to one subunit will affect the binding property of other subunits on the same protein. So that is a definition, basic definition of allosteric protein. So let's find out whether hemoglobin is an allosteric protein or not. obviously we have talked about the cooperative binding. So cooperative binding is a form of allosteric binding. So obviously hemoglobin is an allosteric protein. So here I have tried to draw a hemoglobin structure having four subunits. So we know that hemoglobin firstly is present in T state, that is very unstable when oxygen get bound to it. So when at the first subunit oxygen bind, then it bind it to very weakly, and gradually due to changes in the binding affinity, because the hemoglobin is an allosteric protein, so gradually we are, if we are adding oxygen molecules in this, the conformational or binding properties will get changed, and the oxygen get start to attach very easily, so the R state can be easily achieved here. Thanks for watching.